Hello, hello. This is Holland Chambers Biology bringing to you lecture number 6, which is all about dihybrids. So let's go ahead and get started with some definitions here. Remember, mono in Latin means one, di in Latin means two. So monohybrids was where you guys have been putting together one trait crosses, and that would be, for example, just hair color. Okay, so big H, little four square boxes. Okay, dihybrids, today we're going to be looking at a two trait cross. So instead of just hair color, we're going to do hair color and let's say hair texture. So brown versus blonde, curly versus straight. And then all of a sudden, we are going to be creating, instead of a 4-square grid, we are going to be creating a 16-square grid today. Yay, hands up in the air. Here we go. All right, so dihybrids, because we're going to be dealing with two traits, we also need to have a key that incorporates both traits. So let's go and look at Mendel's P study. In his experiment, he found that yellow peas were dominant over green. So therefore, all you need is a big Y in order to be yellow. So big Y, or big Y, little y, and we will be yellow. The texture of the pea was either smooth, round, or wrinkled. So looking at his original homozygous purebred parents, he got yellow, round peas. So because they were homozygous purebred, and because yellow and round are dominant, we are now going to be looking at 4 letters instead of 2. So one from mom, one from dad, one from mom, one from dad. So 2 letters for color, 2 letters for texture. Now the other parent was green and wrinkled. So green is little y, and then wrinkled is little r, little r. Now it's important that you guys keep color and texture separate. Don't do YR, because when we get to our 16 grid cross, it'll make a lot of sense why to keep them separate. Now remember, when you have 2 parents, each parent can only give half of their DNA to their child in their gametes, and this is referred to as the law of segregation. So mom over here has a big Y. She's got 2 of the same, so we're just going to give 1 to her child. A big R, 2 of the same, so only give half to our child. So she's going to give a Y and an R in her egg. Dad over here can only give half, and he only has a little Y and a little R to put into his sperm. So when you put the egg and sperm together, again, this is where it's important. Don't do YR. Put all the color first. So the color is Y and then little y. And then do this texture second, which would be your R's, big R, little r. So every baby that's created from these homozygous dominant and homozygous recessive parents are going to be heterozygous. Now the question is, what if we were to mate 2 of these babies together? What would their children be? So our original parents are the P generation. What we just created through this egg and sperm are our zygotes, fertilized eggs of the kids, which is your F1 generation. Now I want to take 2 of our children and mate them together and then come up with the F2 generation. So in order to do that, we are going to separate out our different traits, color and texture. We're going to take one allele from one trait, and we're going to connect it to another allele of a second trait. This is actually referred to as foiling. So in one egg, we're going to put a big Y and a big R. Then we're going to do the same thing. A big Y could also connect to the second trait, little r. So big Y, little r. We could also do a little y with a big R. So that will go into another egg. Or we could have the DNA of a little y and a little r in our gametes. Okay, so these are our possible options that we can give to our children. This is all referred to as the law of independent assortment. So independent assortment, again, is that these alleles, despite color and texture, will be inherited independently without influence upon each other. So just because we inherited a big Y doesn't mean that it's going to alter or change the roundness of this inheritance. So we're not going to suddenly become purple and we're not going to suddenly become wrinkled. So Y and R stays as Y and R. Okay, so let's go ahead and take two of these children and mate them together. So on the second page, I've already drawn this out. So again, at any time, make sure you guys just stop the video so that way you guys can write this stuff down. Okay, so the question is, what would our F2 generation be? So I highly recommend that when you guys get a dihybrid, take your parents and slice them in half. So we have two boxes, one trait, two traits. And then you're going to take that first trait and you're going to foil it with the second trait. So big Y, big R. You also have big Y, little y, big R, or little y, little r. Okay, so these are mom's eggs or gametes. Now, because dad over here looks exactly the same as mom, just use the same combination and put it in the sperm. So big Y, big R, big Y, little r, oops, little y, big R, and little y, little r. Okay, so now we're going to drop it down, bring it over. So we're going to connect the egg and sperm together and create a zygote or baby option. And again, please put all the Y's together and then please put all the R's together. Otherwise, it's going to be really hard to read this. So YY and then RR. Okay, so the next baby, drop it down, bring it over. So Y, Y, and in this time, it's big R, little r. Okay, and you just keep doing that. So here's a big Y and a little y. And again, please make sure with dihybrids to always write the capital letter first, so we know what we're looking at when it comes to dominant recessive. The next one is a big R, big R. Okay, so again, color, texture, color, texture. So from here, what you're going to do is that you're going to write out, just like we did with our monohybrids, what all of our data is. So what I recommend is that you start with the first box, YYRR, and you're going to tally it and then cross it off. Okay, how many of those do you have? Go through, count them up. I've only got one. And the second box I have is YYRR. I'm going to cross them off and I'm going to tally. I'm going to go find another one of him and cross them off. So eventually you guys end up creating this nice big genotype. Okay, so if you were to fill the entire thing in, here's all my different tallies of each of these combinations that I have within my 16 square grid. And then from your tallies, make life simple and convert that into numbers. All right. Now, from the genotype, we're now going to be able to figure out what these 16 children are going to be phenotypically. Okay, so now we no longer need our grid or Punnett square. What we're going to use now is just the genotype. So the first thing you're going to do is you're going to think, okay, big Y is yellow, big R is round. How many big Y, big R's do I have? Well, the first one I have one, so we're going to cross it off. We're going to write one. Okay, here's another one, big Y, big R. How many do I have? There's two, so I'm going to do plus 2. All right, cross them off. Big Y, little r. That's not yellow and round. So I need big Y, big R. So here's another big Y, big R. There's 2, so plus 2. Big Y, big R, there's 4. Cross them off, plus 4. Any more big Y, big R combinations? Nope. Okay, so then 1 plus 2 plus 2 plus 4 equals 9. Okay, now let's go down to the second combination. So big Y and 2 little r's. 2 little r's means that we are wrinkled. So that means that we are yellow and wrinkled. Okay, so I have 1. So you do 1 and then you cross them off. Okay, so be okay with that. Just cross it off. Now let's go down, look for another big Y, little r combination. So these two are already crossed off. We don't need to worry about that. Here's a big Y, 2 little r's. There's 2. We're going to cross them off, plus 2. Okay, so there's no more big Y's in this combination. So now add up your total. 1 plus 2 equals 3. Okay, and you go down and you do the whole entire thing. So these are all crossed off. So it looks like we're just going to mess around with these ones here on the bottom. Little Y's are now green. Big R is round. So here's another 2 little Y's and a big R. So 1 plus 2. So 1 plus 2 equals 3. cross them off. And then the very last one we have is 2 little y's and 2 little r's equal 1. So therefore it's green and wrinkled, it's pure recessive equals 1. So here's the kicker. Whenever you have two parents that are heterozygous, right back up here, heterozygous, two parents that are exactly heterozygous for two different traits, you will always get an F2 generation of a 9-3-3-1 phenotype. So 9 being homozygous or dominant, round yellow. You also have, oops, I'm going to take that out. So this is dominant is what I meant to say. So 9 dominant traits. You'll get 2, 3 heterozygous traits, which is yellow and wrinkled or green and round. So this is dominant recessive, recessive dominant, and then you'll get a 1 pure recessive trait. Okay, so again, 9-3-3-1, you'll always get this phenotypically if your parents are both heterozygous. Now, what happens if your parents are not heterozygous? Well, this other parent, if it's not heterozygous, you would just do the same type of foiling with whatever the alleles are, put his combinations here, and then you're going to do the same thing. Cross it off, write out your data, and if they're not both heterozygous, this data is going to be different. Okay, and you're not going to get a 9-3-3-1. You might get a 12 to 4, or you might get an 8 and an 8 with whatever phenotype that you guys are looking at. So remember, take it step by step. Okay, cross it off, write out your data from your genotype, then configure your phenotype, and never assume it's going to be 9-3-3-1. Okay, just always make sure to work with your data to verify. All right, if there's any questions, please make sure to ask in class. Otherwise, this is Hole in Chambers checking out.